What did you say? 23?
It's total is 46 pairs.
46, it's 23 pairs.
Yeah.
So, because the stem cell undergoes mitosis, the type B daughter cell has 46 chromosomes, just like the stem cell. But because it's beginning the process called meiosis, a specialized type of cell division, where the numbers get cut in half, its daughter cells are going to have fewer. Now, meiosis is divided into two parts. With meiosis 1, you produce two. Well, let's talk about this first. This is type B daughter cell. It differentiates into a primary spermatocyte. And the primary spermatocyte has 46 chromosomes. This is the first cell to undergo meiosis. So the first part of meiosis is called meiosis 1, in which you produce two secondary spermatocytes, each of which has now half the number of chromosomes. So this is called meiosis 1. Then these secondary spermatocytes each then undergo meiosis 2. Then we have a total of 4 from the original primary spermatocyte. And each of these daughter cells have a total of 23 chromosomes. In this case, the daughter cells from the division of the secondary spermatocyte are called spermatids. All right, so let's see if we can follow this. Here's your spermatogone again. Type A daughter cell, type B. Type B differentiates into a primary spermatocyte with 46 chromosomes. When it says 2N, that means 2 sets. So 2 times 23 is 46. Now, when this undergoes meiosis 1, we produce 2 secondary spermatocytes, each of which have an N number with 23 chromosomes. Each of these cells completes meiosis 2, giving us a total of 4 early spermatids. And these spermatids each have 23 chromosomes. So from a single primary spermatocyte, we produce 4 spermatids. These early spermatids are also called round spermatids. If you look at them under the microscope, they have a nice centrally located nucleus, and the cells themselves appear rounded and have a lot of cytoplasm. As these spermatids develop, they start to lose some of the cytoplasm, and their shape undergoes transformation. Okay? So if you saw a cell that looked like this, or it may look like this would be then a later spermatid. You may even see it starting to develop a little bit of a tail. So you see something like this. Okay. These would be late spermatids. Okay, and then finally, and by the way, when you see this much cytoplasm, these are late spermatids, and they're going to differentiate into spermatozoa, as we see here. So these would be late spermatids, these would be spermatozoa. And the spermatozoa, the nuclear material is going to be concentrated within the head. Mitochondria are going to be located within a tailpiece. So the mitochondria, remember, are the structures, the organelles, which take the nutrient, whether that be fructose and citric acid, and they transform it into ATP, and then you can have a flagellum that serves as a tail. So the genetic material, the 23 chromosomes, are going to be in the head, mitochondria in the mid-piece or neck piece, and then the flagellum is going to form a tail. The head of the spermatozoa is going to have a little helmet-like structure called an acrosome. And the acrosome is going to contain enzymes. They're called acrosomal enzymes that are going to be used to help penetrate the female ovum to achieve fertilization. Now, you may not have noticed it, but there's a columnar-shaped cell that sits on either side of these developing spermatozoa. Okay, it's pictured here in yellow, and it's got kind of an oblong nucleus with a very prominent nucleolus. This is called a Sertoli cell. You can either call it a Sertoli cell, which is named after, it's a proper name, named after the individual who first described it, or you can call it a sustentacular cell. These are nurse cells that nurse the developing spermatogenic cells. They provide physical protection. They provide the correct chemical environment for these spermatogenic cells to develop. So they help guide the process of spermatogenesis. When I say the term spermatogenesis, I'm talking about the entire process where we start with the spermatogonium and end with spermatozoa. That process is called spermatogenesis. Now, if I simply start with the early spermatids and talk about their development into spermatozoa, the late part of spermatogenesis, that's called spermiogenesis. So spermiogenesis simply goes from the spermatids to spermatozoa, whereas spermatogenesis is the entire process, starting with spermatogonia. Okay, let's talk about transport of the spermatozoa that are developed within the seminiferous tubules. Let's talk about how they get transferred into the female reproductive tract. We said last class that the penis has to become more rigid, hard, in order to accomplish penetration of the vagina so that the spermatozoa can be expelled into the vaginal tract. So, of course, this is called an erection. And an erection is a parasympathetic process, whereas orgasm and ejaculation is sympathetic. You know that the parasympathetic division of the autonomic nervous system is also called the craniosacral division. And so the parasympathetic fibers that are going to be necessary to provide input for achieving an erection are going to come from the sacral portion of the spinal cord. S2, S3, and S4. In medical school, you learn lots of little mnemonics. And so it would be S2, 3, and 4. They said keep the penis off the floor. Okay? And simply a mnemonic to help you remember what segments of the spinal cord are responsible for an erection. S2, 3, and 4. So the act of achieving an erection is a reflex mechanism. It's initiated by stimuli, psychic and or tactile stimuli. Okay? That travel to the sacral portion of the spinal cord and trigger the release of parasympathetic impulses. Now, these parasympathetic fibers travel to the endothelium of the penile arteries and arterioles. Okay? And they trigger the release of a substance called nitric oxide. Nitric oxide is the most potent endogenous vasodilator that the body produces. It's going to cause those penile vessels to vasodilate, and in doing so, carry more blood flow into the penis. Okay. So it's going to increase arterial blood flow into the penis. If we look here, cross-section through the penis here. This would be a cross-section through a flaccid penis, where this would be an artery giving rise to arterioles that are providing blood flow to little sinusoids. Within the erectile bodies, whether it's the carpa cavernosa or corpus spongiosum. Okay? So this is a flaccid penis. We see the sinusoids, and then the sinusoids are emptied into veins that drain the penis. When nitric oxide is released, look what happens to the penile arteries. Compare this to this. The penile arteries and arterioles vasodilate, delivering more blood flow to the sinusoids. So the sinusoids expand with blood. And as they expand, they compress the venules that are there to drain that blood. So that allows blood to flow into the penis, but it blocks its exit. So what happens? The penis becomes more rigid, hard, as it becomes engorged with blood. Now it's capable of penetrating the vagina. Now we said that the act of achieving an erection is parasympathetic. Orgasm, on the other hand, is sympathetic. So when sexual stimulation becomes intense, then we trigger now a sympathetic reflex. Okay. Remember that the sympathetic division is the thoracolumbar division of the autonomic nervous system. So the sympathetic fibers are going to be derived from the lumbar region of the spinal cord, and they're going to trigger peristaltic contractions, waves of smooth muscle contraction, okay, within the ducts of the testes, the epididymis, and the vas deferens. Okay? So what's going to happen? Spermatozoa that are located within those structures are going to be propelled forward into the urethra. Okay? At the same time, sympathetic impulses are going to trigger peristaltic contractions within the accessory glands and their ducts. That's going to propel seminal fluid into the urethra, where it will mix with the spermatozoa. Okay? So we're producing semen here. Now, other rhythmic impulses are going to come from the sacral portion of the spinal cord and stimulate skeletal muscle at the base of the penis. And then that's going to propel the semen present within the urethra to be expelled or ejaculated. All right, let's talk a little bit about hormones. You know that the hypothalamus produces gonadotropin-releasing hormone, which travels via a portal system to the anterior pituitary and facilitates release of gonadotropins. Those gonadotropins are FSH and LH. In the male, LH, luteinizing hormone, targets the interstitial cells of Leydig. Okay. These are those islands of cells located between seminiferous tubules. The way to remember that is luteinizing hormone has an L, cells of Leydig have an L. So it's easy then to remember that the luteinizing hormone targets the interstitial cells of Leydig. And it causes those cells to release testosterone. It turns out that testosterone is necessary for spermatogenic cells to develop into spermatozoa. They have to be bathed in testosterone. Okay. Now, at the same time, FSH targets the Sertoli cells, or the sustentacular cells, and triggers their release of a protein called androgen binding protein. Androgen binding protein is released to the spermatogenic cells where it tends to bind to testosterone, hence the name. Okay? So it maintains, this androgen binding protein maintains the testosterone-rich environment, okay, in and around the spermatogenic cells so that they develop normally. Now the Sertoli cells also inhibit, which provides negative feedback to the hypothalamus and pituitary. Testosterone also provides negative feedback. Okay, so during different parts of life, you see little peaks in testosterone production. In males, during early prenatal development, testosterone facilitates the development of male internal genitals as opposed to female. Just prior to birth, there's a peak in testosterone levels that stimulate the descent of the testes. At puberty, we have another peak in testosterone levels that stimulate the development of the male sex organs and secondary sexual characteristics. And you should know that testosterone stimulates the metabolic rate, so that males tend to have a higher basal metabolic rate than females. We've discussed the fact that it's also the basis for libido. And you might also know that it helps stimulate erythropoiesis, so that in males, generally, your hemoglobin and hematocrit normals are a little higher than in females.